Yeah, you can see on the board, welcome class 12 in the online bio classes. Already we had started the second chapter, that is sexual reproduction in plants. And in our previous class, we have already studied about the flower is the reproductive part of the plant, and flower has different parts, the four different walls of the flower: calyx, corolla, androecium and gynoecium, which has different functions to do. Then we studied some technical terms associated with the flowers, and now today we are going to study about the structure and development of anther and male gametophyte. You know what is anther? Anther is the part of the flower which produces the male gametes called pollens. So you know that the anther lobe is dithecous. Dithecous means each anther contains two lobes like this. So such an anther which has two lobes is called dithecous. Each anther contains two lobes, and inside each lobe there are two pollen sacs. Means total how many? Four will be there. Each lobe has two pollen sacs, means two chambers are there. So 2 plus 2, 4 pollen sacs are there. And this pollen sac is also known as microsporangium. What is it called? Microsporangium. Okay. A large number of pollen grains. So the other name of pollen sac is microsporangium. Many pollen grains, as I can show here in the diagram, these are the pollen grains. Many pollen grains are present in each sac. Like here you can see, this is the diagram of TS of anther, transverse section of anther. If you cut the anther of the flower, in the transverse section you will find that it has 4 chambers. 1, 2, 3, 4. Means there are total 2 lobes. Each lobe has 2 chambers. Like this is one lobe, so 2 chambers. This is another lobe which has 2 chambers. So total 4 chambers are there. And inside these chambers there are pollen grains, which are also known as microspores. So what is the other name of pollen grains? They are also known as microspores. So microspores is the other name of pollen grains. Got it? What is pollen grains? Pollen grains are the male reproductive cells which are produced in the anther lobes. And the pollen sac, this one I had said, no, it is called pollen sac. Pollen sac is also known as microsporangium. What is it called? The other name of pollen sac is microsporangium. Alright. Now next you have to study structure of anther and formation of microspores. What is the structure of anther and how the microspores or the pollen grains are produced. Just try to understand and listen carefully so that you can understand the topic. The diagram which I have drawn, that is of TS of anther. It is there in your syllabus. As a homework you can practice drawing the same diagram. If you have the book, you can take help of your book to draw, but practice labeling them without looking at the diagram. Okay. Now next is structure of anther. A young anther usually becomes 4-lobed at an early stage of development. So here you can see 1, 2, 3, 4. Young anther becomes 4-lobed at an early stage of development. 1, 2, 3, 4 lobes are there. Actually there are 2 lobes, but 4 chambers are there. It's wrong to say that 4 lobes. And some hypodermal cells. Now what is hypodermal? Here you can see in the diagram, the outermost layer of cells is called epidermis. What is it called? Epidermis. Just below the epidermis, the layer of cell which I have marked here, it is called hypodermal cells. Okay, so what is hypodermal cell? The cell layer which is present below the epidermis is called hypodermal cell. So this hypodermal cell, they become distinct. Distinct means they become somewhat unusual, separate, away from the rest of the cells, and they are known as archisporium. The word which I said, remember it, there is no place, otherwise I could have written here archisporium. I will try to rub this one and write. Okay, archisporium. It's very important concept, you must understand that. What is that? Archisporium, or archisporial cells also it is called. Archisporial cells or archisporium. Archisporium. So what is this archisporium or archisporial cells? They are the hypodermal cells, some of the hypodermal cells which become distinct and they behave as archisporial cells or archisporium. Now, the archisporial cell in the next step, they undergo periclinal division. Now, what is the meaning of this word periclinal division? What is the meaning of periclinal division? They divide periclinally. Anticlinal and periclinal, two words are there. Periclinal division means parallel division. Periclinal means parallel division. Like the cells of archisporium, they will divide parallel to each other and they will form two layers of cells. One to the outside and one to the inside. The cells which are produced in the outer layer, it is called primary parietal cells. What is the word I said? Primary parietal cells or primary parietal layer. And the one which is formed by the division of archisporial cells to the inside, that is known as primary sporogenous layer or sporogenous tissue also we can say. So primary parietal layer is formed to the outside and primary sporogenous layer is formed to the inside. Two layers are formed. From where it is formed? It is formed by the division of archisporial cells. Got it now? Up to here you have understood. Now next, the primary parietal layer, it divides again. The one which was formed to the outside. Parietal layer was present in the outside, so it divides again and it forms a number of cell layers which forms the wall of the anther. So you can see the wall of the anther is there. Anther has many walls. Like outermost layer of the anther, number one, epidermis is there. Second layer, it is known as endothecium. This is the second layer from outside. Number one comes epidermis, then endothecium. It is made up of columnar cells. Epidermis is made up of tangentially stretched cells, means the cells which are stretched. Endothecium consists of columnar cells. Then we have middle layers. In the middle layers you can see two or more layers of cells are present. So here I have drawn only two layers. It can be three layers, four layers also. Okay. So two or more than two layers of cells are present. That is called the middle layers of cells. Understood? Then after that we have this one. It is known as tapetum. This layer is called tapetum. I have marked also. This layer is known as tapetum. And it encloses the sporogenous tissue in which the pollen grains will develop. Okay? And what is the function of tapetum? One by one we will study. The function of all these, so you can see from this diagram, the wall of the anther mainly consists of different layers. What are they? Epidermis being the outermost layer. Second is endothecium. Third one is middle layer, which is made up of more than two layers of cells. And the last one is a single layer of cells, which is called tapetum. Only one layer of cell is present. It is the one which provides nutrition to the developing pollen grains. So its function is to give nutrition, food. Because these are pollen grains, they are living cells, they need food, they need to grow and they need to increase in number. For that the food requirement is met by tapetum. Got it now? Now next we have, I was saying about the primary parietal layer. Okay, it gives rise to or it divides to form number of cell layers which forms the wall of the anther. So wall of the anther is made up of what are the different parts? Epidermis, endothecium, middle layer and tapetum. So fourth one I will write this one number 3 and this is the fourth one. Got it? Understood? Now endothecium has columnar cells. You can see I have written also here. Whereas the epidermis has stretched cells and the cells of the middle layers, they are ephemeral. The meaning of the word ephemeral is they are about to die. They will not further work. So cells of middle layers, they are ephemeral. Before the process of meiosis cell division, all these cells will degenerate. They have no functions to do. So outer three wall layers, that is epidermis, endothecium and middle layers, all these number 3. Their function is, if it is asked what is the function of these 3 layers, the answer is since they are present or located to the outside of the anther, so they help in providing protection. And the function of this tapetum layer number 4 is to provide nutrients, that is nutrition to the developing microspores as well as microspore mother cells. Okay, now what is microspore mother cells? tapetum serves as a nutritive tissue for pollen mother cells. Pollen mother cells means from where the pollens are produced, and to the microspores also. So tapetum has two functions: to give nutrition to the microspores, that is pollen grains, as well as to the pollen mother cells. Now what is this PMC? Pollen mother cell. Which cells are called pollen mother cells? It is the sporogenous tissue which was formed. I told you in the beginning that some hypodermal cells, I will draw and show you with the help of diagram also after rubbing this one. Okay, some hypodermal cells which are present in the anther, they become distinct and behave like archisporial cells. Now these archisporial cells divides, the cells will divide periclinally, that is parallelly, to form two layers of cell. The cells which are formed outside, that is called primary parietal layer, and the one which is formed towards the inside is called primary sporogenous layer. So that primary sporogenous layer, the second one which I was talking, it is the one which gives rise to the pollen mother cell. So primary sporogenous tissue may either directly it will produce the pollen microspore mother cell, or it will function as microspore mother cell, or this primary sporogenous tissue will undergo the type of division called mitotic division, and then it will act as pollen microspore mother cells. Microspore mother cell means the pollen mother cell also we can write, because pollen's name is microspore. So MMC also you can write. If MMC means microspore mother cell. Microspore is same as pollen grains only. So I have written here PMC or MMC, they are same. Microspore mother cell or pollen mother cell, they are the same. Okay, they are the sporogenous tissue which gives rise to the pollen mother cells. And from the pollen mother cells, the pollen grains are produced. From these pollen mother cells only, the pollen grains are produced. And this process of, that is also there in your syllabus, just mark it. What is microsporogenesis? So what is the answer you write? Microsporogenesis is the process of formation of microspores. Microspores means pollen grains from the sporogenous tissue or the pollen mother cells. Anyone you can write. In your book it is mentioned from the sporogenous tissue. So process of formation of microspores from the sporogenous tissue is called microsporogenesis. Okay. Now next, I had told you, I will show you that diagram, the one which I was saying. This one hope you have understood. Once again, it's important concept to understand. Okay, because this TS of anther is there in the syllabus, so you must practice drawing and labeling all the parts. Okay. So what are the different parts? Maybe the diagram is not so nice, but you try to practice and draw it more better than this. Now you can see there are 4 anther lobes shown in the diagram. What are they? They are the anther lobes. You know what is anther? Anther is the male part of the flower, which consists of, like, this is the anther part, this is the filament part, and together they form the stamen. Stamen is the male reproductive part of the flower. Inside each anther, because anther has 2 lobes, it is called diathecus. And inside each anther lobes there are pollen sac, which is also known as microsporangium. What is the other name of pollen sac? Microsporangium. Alright. Inside the pollen sac, you can see these are the pollen sacs. What is present? The pollen grains or microspores, which are the male gamete, they are present inside it. Okay. So next, what we studied? We studied the diagram of TS of anther and the structure also. That anther contains, the wall of the anther is made up of different types of cells. Like the outermost layer of cell is called epidermis. I have marked it number 1. Number 2, the second layer of cells which are columnar in shape. As you can see, this is the columnar shaped cells. That is known as endothecium, the second layer of cell. Then third, we have 2 or more layers of cells, which is called the middle layers of cells. And the cells of middle layers, they are ephemeral. Ephemeral means they will degenerate, they will die. They will not remain alive for a long time. Then we have the inner single layer of cells which is known as tapetum. I have marked it here as number 4. The function of tapetum is to provide nutrition or food to the microspore mother cell and the microspore. They give food to them so that they can remain alive. Okay. Now next, this stomium I have marked is the passage-like structure that is called the stomium. Here also you can mark. Okay, and this one in the middle it is known as connective. Okay, so now I'll rub it. Next we will study about the diagram which I was saying about the archisporial cells. The same diagram only, little bit modification is there. And then we have to study in our syllabus about the pollen grain, pollen viability, structure of pollen. So many terms are there. Okay, so next I had told you, like see, this one is the structure of anther. Now this anther consists of many cells in it. See, all these cells are present. I'll just fill it up. Different stages of development of male gametophyte. Suppose this is A. Okay. Now this layer, the outermost layer is called epidermis. This outermost. Just below that, this layer of cell is called hypodermis. So some of these hypodermal cells. Hypodermal cells. This layer, outermost layer, it is called epidermis. I told you I will show you with the help of diagram so that you will understand. So epidermis means outermost layer of cell. And just below that, the layer which is present, like this one you draw straight, this is epidermal layer. Now below that, if I will draw any layer of cells, like this I am drawing different shapes so that you can understand. This layer is called hypodermal, just below that. So these hypodermal cells, they will become distinct and they behave as archisporial cells. Archisporial cells. So it is the hypodermal cell only which become distinct and behave as archisporial cells. Got it? Now next, these archisporial cells in the next stage, that is number B, they will divide. Okay, what type of division is there? Parallel division. And it will form different types of, like if I draw this thing. If I draw the outermost epidermal layer first, like this, epidermis. Now, I will draw the cells which represent like this one, with the nucleus. Rest all just fill it up, the cells. We have to mark this one only. Now, division of archisporial cell took place, and this one only archisporial cells. Now, their division will take place. So, this is number B. Next, in number C, you will find that two layers of cells are formed. So, I will try to draw it like this. Suppose this is the outermost layer of cell. Again, two layers of cells are formed. Okay. Now see, outer layer of cells. Suppose here outer layer of cells are formed. Inner layer of cells are there. Now this outer layer of cells which are formed, see by division, this outer layer they are called primary parietal layer. What is it called? Primary parietal. And the one which is formed towards the inside, like these are the inner cells, that is called primary sporogenous layer. So what is this primary parietal layer, primary sporogenous layer? These two are the layers of cells which are formed by the division of archisporial cell. Got it? Understood? Now which one is to the outside? The parietal layer is towards the outside. These two are the parietal layers. They are outside, and this one is located inside. So that is called the primary sporogenous layer. Understood now? Now in the next diagram you will find that epidermis, just like the diagram which I had drawn in the TS of anther, it is similar to it. So there will be epidermis, there will be endothecium, which is formed, the different layers of the walls of anther will be formed due to division of this primary parietal layer only. Okay, this primary parietal layer will divide, it will form a mass or a layer of cells that forms the wall of the anther, like endothecium will be formed from it, and then we will have the middle layers formed from it, then we will have the tapetum, and finally the pollen grains or the microspores. Okay, got it? Hope you have understood up to here. Now in the next topic which we are going to do is what is microsporogenesis. I hope I have said that. What is microsporogenesis? The process of formation of microspores from the sporogenous tissue. This is the sporogenous tissue. From here only the microspores are produced. And this one was produced from archisporial cell. And archisporial cells are nothing else but the hypodermal cells. And hypodermal cells are located just below the epidermis. Understood? Now you have seen that the pollen tetrad and structure of microspores. The 4 microspores which are formed from a microspore mother cell. Each microspore mother cell produces 4 pollen grains. You know that by the meiosis cell division 4 daughter cells are produced, but the chromosome number is reduced to half. So similarly here also the process of formation of 4 microspores from one microspore mother cell, that is known as process occurs due to meiosis cell division. Now you know that 4 microspore, pollens are formed. The way they arrange, there are different modes of arrangement. Like the number 1 which is important is tetrahedral. Now you can look at the diagram which I will draw. In tetrahedral, tetra means 4. Okay, but if you see the diagram, it will appear like only 3 pollens are shown here. 1, 2, 3 are shown. This is tetrahedral arrangement of pollens. How? Because in such an arrangement, all the 4 microspores or the pollens are arranged at the 4 corners of a tetrahedron. And since it is 3-dimensional, if we look at it from an angle, only 3 cells are seen, only 3 pollens are seen. The 4th one is just behind it, and that's why it is called tetrahedral. What is it called? Tetrahedral. What is this tetrahedral? It is the mode of arrangement of pollens. So pollen tetrad means how the 4 pollen grains are arranged. Number 1 is tetrahedral, and the second one is isobilateral. Iso means same. Isobilateral means I will draw it here only. Suppose here there are 2 pollens, 1, 2. The next 2 is also like this only arranged. This type of arrangement of pollen grains, in which all the 4 pollen grains are clearly visible, is called isobilateral tetrad. What is it called? Iso means having both equal sides, means on both sides there is equal number of pollen grains. Similarly, there can be T-shaped, there can be a linear pollens arrangement also like this. In a straight line, all the pollen grains, 4 pollen grains can be arranged. This is called linear. It can be in the shape of T-shape. It can be in the form of decussate. In decussate, what happens? 2 pollens are there like this, and 1 pollen is at the like this type of arrangement. Decussate it is called. Okay. Now next we have, after this, structure of pollen grains. So next we'll do the structure of pollen grains. Okay, now next topic we have to do is structure of pollen grains. You know what is pollen grains? They are the male gametophyte, and each pollen grain is of different shape and size. Generally the pollen grains are very light in weight because they have to be carried away by wind and other agencies like insects, water, or by animals from one part of the flower to the other, or from one plant to the other in cross-pollination. So what is the structure of pollen grains? Each pollen grain consists of 2 types of nucleus, or we also say it as cell. The bigger one is called vegetative. It is known as vegetative nucleus. And the smaller one is called generative nucleus. What is it called? Generative nucleus. Then it has 2 layers. The wall is made up of 2 layers. This is the inner layer. Inner layer is called intine. The word which is used, inner layer is called intine, and then outer layer it is known as exine. This outer layer is called exine. Outer layer, it is called exine. Got it? Apart from that, the cytoplasm is there, and the other materials which are present in the cell, general plant cell, it also contains. So there are mainly two types of nucleus: vegetative nucleus, generative nucleus. This generative nucleus, this will divide to form two male gametes or the male sex cells at the time of fertilization. Understood? So male gametes are generally non-motile, means they cannot move on their own. So the male gametes here, they are transferred with the help of pollen tube. Okay, from the anther, from the stigma part of the flower to the ovary. Now let's talk one by one about the size and all. First one is the pollen grains. They generally measure 25 to 50 micrometer in diameter. The size of pollens, 25 to 50 micrometer in diameter. The size of pollen grains. What is the size? 25 to 50 micrometer. What is the size of pollen grains? This one, 25 to 50 micrometer. Means it's very small in size. It generally has two layer wall. The outer layer is called exine, and you may be surprised to know that this exine is made up of a substance which is called sporopollenin. What is this sporopollenin? The one which makes exine. No enzyme is known to us in this world which can dissolve or which can spoil sporopollenin. So that's why pollen grains are found in the fossil forms, because they remain safe from any type of physical or mechanical injury. Even acids, higher temperature cannot damage the pollen grains due to the presence of sporopollenin, which forms or which makes the exine layer, that is the outer layer of pollen grains. They are so tough and strong, which makes them viable for a long time. The second layer is intine. Intine is soft and it is made up of the substance which is there. The substance are pectin and cellulose. Pectin, you know, pectin and cellulose, both of them are types of carbohydrates. So they makes the inner layer, that is intine. It is made up of pectin and cellulose. Cellulose makes the cell wall also. And now, what is germ pore? Germ pore means the tiny pores which are present in the exine through which the pollen tubes are formed. Like from here, the pollen tubes are formed by, this is called a pollen tube. They are formed from the tiny pores which are present. They are known as, these pores are known as, suppose here also I will draw a pore. This pore is called germ pore. Germ pore. Pore means the gap. So what is germ pore? It is the gap which is present in the wall of pollens through which the pollen tubes will arise. This is pollen tube which carry the cells, that is the male gametes, to the ovary part of the flower. Got it? Understood? Now next, pollen grains are densely cytoplasmic, means they contain plenty of cytoplasm, and once they are released from the anthers, they reach up to the stigma of the flower. But most of them, they by accident, they fall in those places which are not suitable for them to germinate, so they are spoiled. Particularly in case of air pollinated and water pollinated flowers, they produce plenty of pollens. Why? Because most of them are wasted. Only few of them are able to reach on the stigma of their species of plants and bring about fertilization. So now next you have to study is economic importance of pollen grains. It is there in your syllabus. What is the economic importance? Why pollen grains are economically very important? If you see a flower, the powder-like substance which comes from that flower, they are the they are the pollen grains. So these pollen grains are released from different types of flowers into the atmosphere, causing different types of breathing or respiratory problem. It also adds to the air pollution and brings about bronchial implications like asthma, bronchitis, and so many diseases are caused. Like allergic reactions are produced due to these pollen grains which are present in the atmosphere. So pollen grains here, they are the harmful effects of pollen grains. No, pollen grains here we are showing the harmful effect that they are causing disease due to the presence of pollens in the atmosphere which causes pollution. But what is the useful effect or usage of pollen grains which is beneficial for us? It is found that pollen grains are highly nutritive. Nutritive means they provide a lot of nutrients. So pollen tablets are available in the market, and it is assumed means we believe that if a person takes pollen tablets, particularly it is good for his body power, and particularly for the athletes means those who are running races, it can increase their stamina if they consume the pollen tablets. Now next is viability of pollen grains. This is also there in your topic, viability. Viability of pollens. The meaning of viability of pollens is generally pollens are shed from the pollen tubes, like from the anthers the pollens are released by the dehiscence. Dehiscence means splitting of anther. The pollens are released into the atmosphere and they have a particular lifespan. Lifespan means the time up to which they can remain alive. Some examples are there. I will cite that example also. That is called the viability of pollens. So example is in case of rice and wheat. You know rice and wheat are the plants. When the pollens are discharged, they remain viable. Viable means alive. They have the capacity of fertilization only up to 30 minutes after being released. Like if they are released now at 9 a.m. till 9:30 they will remain alive. After that they are not going to bring about fertilization. But there are some other examples also given, like in case of Solanaceae family, that is potato family, in case of Fabaceae family, the pollens can remain viable for many months. Like today if they are released from the flower, they will have the power of reproduction or fertilization up to many months. Like in this month if they are released, that is the month of June, up to August, September, October, November, December also they can bring about fertilization. It means they have a longer period of viability as compared to the other plants. Got it? Now nowadays, in order to save our time, the pollens which are genetically one of the important part of the flower, male part of the flower, they are preserved because they are the male gametes. So they are preserved. Preserved means kept at a very low temperature in liquid nitrogen. The temperature is minus 196 degree centigrade. At such a low temperature in liquid nitrogen, the pollens, live pollens, means the pollens which are good or working condition, they are preserved for future use. Whenever we need, we can use them to bring about fertilization. They are stored in the pollen bank. Just like we have the blood bank, pollen bank is there where we store the pollen grains from different varieties of flowers and use them according to our needs or according to our requirements. We can use those pollen grains for our benefit, particularly in this artificial this pollination technique. We human beings are the one who transfer the pollens according to our needs. Understood? So this will help in plant breeding also. We can improve the variety of plants by using the pollen banks. So today up to here only. In the next class, now what is the homework I will say? You just try to draw the diagram of TS of anther, and if you can, then explain the structure of anther along with the male gametophyte, development of male gametophyte. Hope you can do it. Okay? Thank you.